Hello, I'm Hong Tin from National Taiwan University. The topic I'm going to share with you today is the formation mechanism of corolla tube. What is the corolla tubes? The corolla tubes is a tubular structure among flowers composed of fused petals, which includes the nectaries and other reproductive organs. Over 1/3 of angiosperm species has flowers with corolla tubes, including Syzygia, Convolvulus, and Corinthia. This is a common flower structure among angiosperm. Why do so many species have a corolla tubes? Scientists have found that possessing the corolla tubes is a successful reproductive strategy to plants, since it establishes pollination specificity, thereby bringing about co-evolution relationship between pollinator and plants, and further generate a variety of corolla tube shapes that drive rapid plant speciation. Pollinators of corolla tubes plants including hummingbirds, songbirds, and moths. Therefore, botanists are eager to understand the formation and development of the corolla tubes at a molecular level. By identifying critical factors of corolla tube formation, they can better understand the important group in plant evolution and further facilitate the flower breeding industry. To understand the mechanism of corolla tube formation, botanists use monkey flowers as material, compare genes and morphology between wild-type and mutant flowers that cannot form a corolla tube. Morphologically, they found that the base of the petal primordia quickly expanded laterally and the interprimordial region also grew caudinary, connecting the initial separate petal primordia. However, they observed that no lateral expansion at the base of the petal primordia in the mutant flowers. The aspect of gene they found that is due to auxin, the hormone that controls plant growth, malfunction preventing flowers from normal growing process. Further, they found that the auxin malfunction is because the irrigation of tRNA off pathway. tRNA is a small RNA molecule that can degrade the precursors of auxin response factor, so-called AF, the transcription factor that negatively regulates auxin expression. When tRNA can inhibit AF, it allows for the synthesis of the corolla tube. However, if tRNA is unable to inhibit AF, the synthesis of the corolla tube may be disrupted, leading to mutant flowers that are unable to develop these structures. Hence, scientists have proposed a hypothesis that auxin expression can affect the growth of interprimordial region and petal primordia. To test this hypothesis, they use gene transformation to make auxin in the petal emit red fluorescence. This allows them to directly observe the disturbance of auxin expression and understand its impact on petal development. The results show that the fluorescence signals in the mutants were generally weaker compared to the wild type, particularly at the gap between the petal primordial base and the interprimordial region. Almost no auxin fluorescence was detected in this area. To confirm such findings, the scientists immune developing flowers directly into auxin and observed that the mutant flowers were able to form a normal corolla tube. This result provides evidence that auxin regulation is important for the development of corolla tube. In the future, the scientists are interested in understanding whether there are different developmental and regulatory mechanisms between different shaped corolla tube. The story of corolla tube is still being continued. Thanks for listening. Goodbye.